Welcome back, and welcome especially to those of you who have only just joined us for this continuation of our program, the conference on the Seafood Tomorrow project. We will have this afternoon 3 presentations on the rapid screening methods for toxins in seafood, and I would like to ask Monica to start first with her presentation on tetrodotoxins. Monica, you have the floor.
Okay, thanks so much to everyone. My name is Monica Campas. I'm a researcher at IRTA. I'm going to talk about the task that we have been performing in the Seafood Tomorrow about the screening methods for the detection of different analytes. So I'm going to share my screen. Let's see if everything works well. Okay, I hope this is going to work. Can you see my screen? Yeah?
Yes. Thank you.
Okay, thank you. So, as I said, we have been working on different screening methods for the management of seafood production systems. In this task, there were different institutions working. We have developed, as I said, 3 different screening methods. One is an enzymatic inhibition assay for the detection of chemical contaminants, and this has been performed by AFTI, who is in Spain. Then the optical biochip for the multiplex detection of regulated marine toxins, and in this case, this has been performed by Queen's University of Belfast in Northern Ireland. And then finally, the immunoassay for the detection of tetrodotoxins, also in seafood. And this has been performed by IRTA, my institution, in collaboration with the Queen's University of Belfast. Additionally, I have to say that the company Biorex Food Diagnostics, also located in Northern Ireland, have been helping all of us, in fact. So I'm going to talk about the immunoassay for the detection of tetrodotoxins, and after me, my colleagues will talk about the other 2 screening methods. Okay, so regarding tetrodotoxins, tetrodotoxin is a potent neurotoxin that was discovered in Japan in the pufferfish a long time ago. And in fact, you can see here the molecule is a very small molecule. And in fact, the tetrodotoxin has this structure, but additionally, we can also find different analogues with different structures, very similar, but with small differences. And we have already identified more than 30 different analogues. And these tetrodotoxins have been responsible for many food poisoning incidents, usually associated to the consumption of pufferfish. And why? Because this tetrodotoxin block the voltage gate sodium channels that you can find in the cells, and this can result in neuronal, respiratory, gastrointestinal effects, and sometimes, unfortunately, even death. So, as I said, tetrodotoxin was initially found in pufferfish, but in the recent years, tetrodotoxin has also been found in shellfish in Europe, in fact. Everything started in Spain in a toxicity episode that people got sick because of the consumption of snails that were contaminated with these tetrodotoxins, although the shellfish was from Portugal, but since then, tetrodotoxin has been found in shellfish in England, in Greece, and also in the Netherlands in 2016. In fact, this was a quite a controversial episode because the Netherlands they decided to close the production areas, the shellfish production areas. It was a little controversy about that. Also in Spain, in Italy, and in France. I have to say that the contents that have been found in shellfish are quite low. So it seems that it's not a problem right now, but it may be. So this means that we have to be ready to have screening methods available for the detection when needed. Okay, so at the moment, tetrodotoxin in the shellfish is not regulated in Europe. So what we have been doing in the frame of the Seafood Tomorrow project is develop an assay based on antibodies. As I said, we are collaborating with Queen's University of Belfast that has provided us with these antibodies. And why we have decided to choose this strategy? Because they are very fast, they are quite cheap, the sensitivity is very good. Additionally, we have to say that the antibody is very, very sensitive, and they are quite simple and easy to implement. And we have been using these antibodies in combination with magnetic beads. And why magnetic beads? Because they improve the assay kinetics and they are very easy to manipulate, and also you can reduce the matrix effects. And matrix effects means the effect that the shellfish, I mean the oyster, the mussel, or the razor clam may have on the response of the antibody. So in fact, we have been working on two different steps of this, the whole process, that are the extraction, where you extract the tetrodotoxin from the shellfish, and the immunoassay, when you detect this tetrodotoxin that you have extracted. So regarding the extraction process, we have been following a protocol that is quite harmonized, and it's quite simple. You just have to prepare an homogenate of your shellfish. You mix it with acetic acid and water. Then you vortex mix, you boil for 10 minutes, you vortex mix again, you centrifuge, and then finally you have to filtrate the extracts. Regarding the immunoassay process, in this case, we can, in a very short way, we can say that you have three different steps. In the first step, we have to prepare magnetic beads tetrodotoxin conjugates. It means that you immobilize tetrodotoxin on magnetic beads, and then you will perform a competition between the antibody and free tetrodotoxin that will be present in the sample, if any. And the antibody will have to choose if it goes to the tetrodotoxin that is on the magnetic bead or the free tetrodotoxin. After the washing steps, you will have a second step where you will add a secondary antibody that is labeled, and this label will give you a response. In our case, a color. Okay. So in this case, I have to say that the response will be inversely proportional. So if in the sample there is no tetrodotoxin, everything will be blue. On the contrary, if there is a high amount of tetrodotoxin, the solution will be transparent. So after this, you can take your samples with the different colors depending on the amount of tetrodotoxin that there is inside the sample, and you can measure the absorbance, and you will have an idea of the content that is present into the shellfish sample. So using this strategy, we obtain very nice calibration curves. Here you can see, for example, the response at different doses of tetrodotoxin. And you have to know that our limit of detection was very, very low, 0.5 nanogram per mil. This is because the antibody is very good. Additionally, we test if we could, when you do the extraction, you can also be extracting other toxins like PSP toxins, paralytic shellfish poisoning toxins, and we observed that our assay is very selective, and it can only detect the tetrodotoxin without interferences from these other toxins. In order to make a very, very simple assay, we evaluate the possibility to do this conjugate, the tetrodotoxin immobilized on the magnetic beads, and evaluate if we could store it and to have it ready for the day that you want to do the analysis. And we observe an excellent storage stability of the conjugate. Additionally, we try to make all the steps as short as possible, and finally, we saw that we could perform the assay in just 70 minutes. So this is very good. So in one morning, you can have your shellfish and have the results. And then we decided to start with analysis of samples. At the beginning, we started with mussels that we spiked with different levels of tetrodotoxins to see if our system was able to detect these tetrodotoxin levels. And here you can see, for example, the 3 different levels, and we could detect tetrodotoxin in all of them. So in our case, for mussels, the limit of detection was 3.3 micrograms per kilo. We are very happy about the result because it's quite low. And as I said at the beginning, tetrodotoxin is not regulated, and there is no regulation about which limit we have to be able to detect. But in any case, the European Food Safety Authority has provided like a guideline value that says that that itself is contaminated with 44 micrograms per kilo, this will not cause any adverse effects on humans. So as you can see, our limit of detection for mussels is lower. So we are happy that we will be able, if needed, we will be able to detect these 44 micrograms per kilo established by EFSA. We have performed the same experiments with oysters. Here you can see the example. In this case, we were able to detect even lower amounts of tetrodotoxin. And here you can see that our limit of detection was 1 microgram per kilo. And the same happened with razor clams. So after all these experiments where we addition the tetrodotoxin, we also performed some experiments with naturally contaminated samples that came from the Netherlands. As I said at the beginning, there was a problem in the Netherlands, so we had some collaborations over there. So they provided with some samples, 3 oysters and 3 mussels, as you can see here. And we compared our results with those obtained with other techniques like LC-MS, instrumental analysis technique, and we could detect tetrodotoxin in all of them. Well, all this information is explained in detail in a publication that belongs to the special issue of Food and Chemical Toxicology. And now I'm going to ask my colleagues to put the video that we have prepared. Okay, can you hear me again? Okay, well, I have been looking at the chat and I have seen that I may be wrong, so I have to apologize the Portuguese people because it seems that maybe it's not proven. No, thanks for the information that the first identification case that was in Spain, maybe the shellfish was not from Portugal. Sorry, Portuguese. Okay, so I share the screen to finish my presentation. Yeah, so I'm almost finishing. In fact, I have to say that Biorix Food Diagnostic is preparing like a prototype. And here you can see the leaflet that they have prepared, where you can see the different shellfish that we have tested and the key benefits, so the conclusions of this work. As I said at the beginning, in 70 minutes you can have the assay as the fastest assay time on the market. The inter and intra-assay precision that I have not talked today, it was too much detail, were really good. And the limits of detection, as I said, 1 ppb for oysters and razor clams and 3.3 ppbs for mussels were very good and appropriate for the detection that we will need to perform if we need in the future. And here you can see, well, it's very small, but they have prepared also the different steps to perform both the extraction process and also the detection with the antibody and the magnetic beads. As you have seen, it's quite simple. You just need maybe some practicing when you are pipetting and the solutions when you have the magnetic beads, but it's easy to learn. And that's it. Thank you so much. And now my colleagues that have been working on the other screening methods will explain about them. Thanks so much. So I'm going to stop sharing. That's it.
Is given that there is no EU legislation for TTX.
Wait a minute, please, Monica, because we have a question on the chat.
Yes.
You see it, or you want me to read it?
I can read it. Who do you anticipate will use this test given that there is no European legislation for tetrodotoxin? Of course. This we have to be ready. I mean, of course, nobody right now maybe the shellfish producers will not be interested in using the kit because it's not regulated. Although I think that it's also good to screen the samples to be sure. Even it's like a plus, that's something that it's good for your company to say that there is no tetrodotoxin. But in any case, we cannot wait to develop the methods and to validate the methods when the legislation will be on. I mean, you have to be ready, and this is what we have been doing for the Seafood Tomorrow project. We have the kit ready in case, and it may happen, tetrodotoxin starts to be legislated and tetrodotoxin starts to increase, the content starts to increase in European shellfish. We have to be ready. That's why we have been working for that. In any case, let me tell you as well that, as I said, we have also been working in the task in the biooptical chip for the screening of the regulated marine toxins that Katrina Campbell will talk about that.
Thank you very much, Monica, for this presentation. And thank you for answering that question. I think that you fully answered the question from Neogen. And I'll ask Katrina if she's ready to take over and move on. Are you here, Katrina? It should work right now. Great. Hello. Good afternoon. Yeah, yes, do. Please try to share your screen. But if you have a video, it may be more challenging. If you could possibly send it so that we can do the sharing for you. Otherwise, we won't hear the sound if you share the videos from your computer. So you have to send it to us so that we can play it, so that everyone hears the sound.
Okay, thank you. So I'm Katrina Campbell. I'm from Queen's University in Belfast as part of the Institute for Global Food Security. And as Monica has said already, we were developing an optical biochip for the integration of fast screening methods for the detection of marine biotoxins, and mainly those regulated toxins. Now, what we've done previously is we've actually mapped out the bivalve supply chain, and our ambition was to be able to put a test anywhere along this supply chain, from source right through to consumers or customers, for them to be able to do their own testing for marine biotoxins. So it was really to allow both regulators, the industry, and what we would describe as novice end users the opportunity to be able to do their own testing for their own management decisions within the supply chain. So we looked at a particular technology for multiplexing, which we'd used in the laboratory before. And this technology from MBio, now called Lightbeck Diagnostics, was one that really caught our attention in its ability to be able to do multiplexing. It also offered a low-cost analysis solution. It's fairly simple to use. It can be highly specific. It offers bespoke sensitivity to get to those action levels required for regulated toxins. And it's also, as you can see from the size of it, it's field deployable in terms of its capability for use. And in the picture, you can see the reader and the cartridge. And really, all the scientific know-how is built into the cartridge. So the end user doesn't need to know all the molecular biology or chemistry as outlined on the slide in terms of antigen-coded or antibody-based assays. The scientific know-how is built within the cartridge, and the reader will give the response in terms of a positive and negative sample. And we have actually looked at this technology at the molecular level for pathogen and speciation testing, protein level for allergen testing, for example, and the residual level for low molecular weight compounds, such as our marine biotoxins, antibiotics, and different chemical contaminants, which might be in the food supply chain. So for marine biotoxins then, this immunological methodology is based on a competitive inhibition assay. So what we do is we immobilize onto our cartridge some toxin coupled to proteins, and then we use antibodies to be able to detect the toxin on the cartridge. And then we use a labeled antibody as a marker to determine presence or absence of antibody binding, which tells us whether or not we've got a negative or a positive result. So the toxins are immobilized onto the surface. We mix our sample and antibody, and we add this to the cartridge. We tend to have an incubation step with the secondary label. Then we place the cartridge into the reader, and there's light directed to the bottom of the cartridge, which allows excitation of fluorophores of the labeled antibody. And this sensitive detection of binding events on the cartridge then can be observed with the reader. And when toxin is present, there is a reduction in antibody binding to the surface. So if we look at the small graph, we get a high signal when there's no toxin present in the sample, and we get a low signal when we do have toxin in the sample. And you can see here, this would be an image, a photographic image of the cartridge. And you can see where the circles are, the red circles are labeled when we have no saxitoxin as one of the regulated marine biotoxins. We get a nice bright response in terms of our fluorophores. And when we have 10 nanogram per mil of saxitoxin, you can see that that signal is completely diminished in terms of we get a very low response to indicate we've got a high toxin concentration. And for this particular system, because all the know-how is actually built into the cartridge system, it's very simple for an end user to use. They don't need to know the biochemistry behind it, molecular biology. They just need to know how to prepare the sample, add it to the cartridge, and then insert the cartridge into the reader and take a read time 5 or 10 minutes thereafter. So we think this is an important to implement these simple testing regimes to allow food business operators to be able to perform their own testing. And it's a simple device requiring minimal sample preparation, either through simple fluid application or dissolution of solid foods and buffering reagents. And you can see here the layout of the cartridge then that we are with the ambition of completing. We've got saxitoxin immobilized as one of the key regulated paralytic shellfish toxins and family, ocodaic acid and the dinophysis esters, domoic acid as a ASP. We've got azospiracid, we've got tetrodotoxin, and these would be the main toxins of concern then within regulations or emerging regulations for marine biotoxins. And depending on the complexity required, we can give a qualitative response, a positive or negative answer, or if there's some level of quantitation required, we can also provide a concentration in relation to the sample that's being tested to give a little bit more information to the food business operators to allow them to make their decisions. Now, we've shown this multiplex detection in harmful algae as shown here, and we've published this work. We have also been able to achieve very low levels of sensitivity in terms of nanogram per mil, whereby in samples we were able to actually detect the toxin by the immunoassay method before it could be detected with HPLC or mass spectrometry. So this gave us a very good starting point then to look at toxins in shellfish. So for the regulated marine toxins, we have saxitoxin antibodies and neosaxitoxin antibodies with different profiles and how they detect that whole family of toxins. We've got ocodaic acid antibodies, and again, how they detect relative to the dinophysis esters to ensure we've all coverage of all the regulated toxins, and antibodies as well then for domoic acid. We also have antibodies for azospiracid and antibodies available for tetrodotoxin. And as Monica has already mentioned today, antibodies are very useful for rapid diagnostic devices because they can limit the matrix effects that might actually be observed with the different shellfish that are being tested. So we get high sensitive antibodies that can be used in these assays. So we look first of all at saxitoxin analysis in shellfish. We devised a very simple extraction procedure whereby we take 1 gram of shellfish homogenate, we add 4 mils of sodium acetate buffer, we vortex it for 10 seconds, we put it through a pH filter bag, and we dilute the supernatant 1 in 250, and then we add it to the cartridge. And we were able to get a very good toxin profile, very much replicating what could be determined by the mouse bioassay in terms of toxicity. And when we looked at matrix effects with different shellfish, we could see because of this dilution factor, we had very little matrix effects present at all. So the saxitoxin assay is very much ready to go on the optical biochip. And when we looked at real samples against the mouse bioassay and against the HPLC method, we had very good correlation across the methods. The next toxin we looked at then was azospiracid, as one of the sort of more regulated, but still emerging in different areas. And again, what we tried to do was to devise a very simple extraction procedure that could be used for this toxin. And as many of you know, probably for marine biotoxins, we have got very lipophilic and very lipophobic toxins. Saxitoxin being more soluble and water-based, and the likes of azospiracid then needing some more solvent in terms of. So in this one, we looked at 90% methanol, and the mixture, again, vortexed, diluted, and very much quick centrifugation, or the page bag, again, that we can use to do the filtering, and a 1 in 10 dilution and assay buffer. And with this particular assay, we were able to get very low sensitivity in terms of nanogram per mil. We're detecting down to 4 or 5 nanogram per mil. But what we did see, because of the methanol extraction and because of the lower dilution factor, a little bit more matrix effects. But when we actually looked at muscle samples, we did see where we had high levels of azospiracid 1. We were able to detect these in the assay. And again, we were able to determine positive and negative samples in mussels. And the same with oysters. We were able to detect very correlating levels of when toxin was present to when toxin was absent. In relation to all the different analogues of azospiracid, there's some work we need to look at in terms of correlation with all the congeners as opposed to just with azospiracid 1. This is very much one of the only methods currently really available for the detection of azospiracid, which is a regulated toxin in the environment. And really, you know, to be able to determine between no toxin present and toxin present is kind of a key area for the regulators. The other toxin we looked at then is the emerging toxin, tetrodotoxin. And then, as Monica has very much alluded to, the background behind this, EFSA very much published their 44 microgram per kilogram equivalent, and it still does require a rapid screening test. Part of the problem with legislation is they're very reluctant to implement legislation if there's no rapid methods or no assured methods for testing. So sometimes the testing methods have to come first before the regulations. And for tetrodotoxin analysis, we're very much able to use this exact same extraction procedure as the paralytic shellfish poisons for saxitoxin. And the dilution is 1 in 4. With oysters, we had minimal matrix effects, and with mussels, we had slightly more matrix effects. But we also looked at this with samples from the Netherlands, and we had very good correlation between our optical biochip assay and the LCM-SMS assay. For the biochip, we got slightly higher values, and again, there's only tetrodotoxin available as an analytical standard. And as Monica has alluded to already, there's greater than 30 different analogues for these different toxins. So the antibody can be picking up some of the other analogues, which perhaps the mass spec wasn't actually determining at that particular time. So a very good assay for tetrodotoxin there as well. And very simple extraction procedure. For tetrodotoxin, we actually validated this method in terms of repeatability, reproducibility, stability, recovery, limit of detection, coefficient of variance. And you can see we get clear separation between the different populations or different toxin concentrations we examined, 0, 10, 20, and 30. So we can clearly differentiate between 0 and 30 nanograms per gram. And we fairly much have a limit of detection, and correlates very much with the ELISA-based type method that Monica mentioned. And the same with the oyster validation. We get very low levels of detection as well for that, fit for legislation for when it should be implemented. In terms of recovery was good, particularly at the higher levels. The percentage CV, there's a little bit of work to be done in repeatability, but again, not bad for a rapid assay that is still being developed in certain ways. So really, in conclusion here, the technology has successfully been used for the detection of regulated and emerging toxins in algal and shellfish samples. We have single-plex assays for each of the regulated toxins. We have a single-plex assay for the emerging toxin, tetrodotoxin. The technology has been evaluated and proven fit for purpose for shellfish samples, and the technology has been evaluated and proven to be fit for purpose for validation requirements, which is also key for the detection of marine toxins as part of the regulation requirements of any new method being applied. And we are still working to combine all the methods so that we can actually, by the end of the project, we are hoping to have all of these toxins on the one biochip and being able to detect them in a single assay, which we're just trying to finalise at this point in time due to some restrictions with COVID. But that's very much our ambition. I'd like to thank you for listening. And I'm not sure if I can share a video, but I'm going to try and share a video of
it depends. Have you got any sound in your video?
There's no talking required, so it should be okay.
Okay, so you can try and let's see.
Okay.
Thank you.
Oh, no, don't do that. So really, this image just shows you the nano plotter actually printing out the slides and adding the toxin. This is the building of the actual cartridge itself. And bear in mind, the end user doesn't need to know how it's printed or how to build the cartridge in terms of their use. You can see the cartridge and the size of the cartridge in terms of where it goes. So this is the part the end user would use. They would really just be running the assay from when they've got their sample and their antibody. They mix them together and they add them to the cartridge surface. And then they would add the secondary antibody, which is the labeled antibody, to the cartridge. And all the biochemistry, molecular biology goes on within the cartridge. And then the cartridge is very much just positioned into the reader. And the results then come up in the form of an Excel spreadsheet that we're working to simplify. And you can see then the variety of spots and spotting that can be done in relation to where the different toxins would be. And the toxins would be spatially defined on the cartridge, so you can identify which toxins are present depending on how illuminated the cartridge might be at that time. And of course, a high signal means no toxins present, such as in these two controls. And a low signal means then when the spots are diminished, that you do have toxin present. So that's just an example or a demo of how the actual system comes together, how it actually is used, and the results that you can see thereafter. So I'm going to stop sharing now, and I'm happy to field any questions.
Thank you, Katrina. Doesn't seem to be any question. They might come in later. So please stay with us, stay online so that you can possibly answer a question that may come after Alex's presentation. Thank you so much anyway for your presentation. Alejandro?
Alex, if you're online, can you please request to speak? I can see you there online.
Everybody, I will share. Let's see. So, my name is Alejandro Barranco, and I am a researcher at ASTI, a research center in Spain. And I will continue talking about fast screening methods as my previous colleagues, in particular those that have been developed within Seafood Tomorrow project for the detection of environmental and processing contaminants. This is the structure of my short talk, I will start with a brief introduction about the contaminants targeted in the project and the importance of their detection in seafood. Then I will explain our approach for their detection, and I will show a video where you will see how the measurement protocol is. Let's see which are our target contaminants. Our work has been focused on three main groups of organic compounds created during industrial processes, including some processes that can be occurring in the food industry. Or maybe they can be synthesized to use in several devices and objects. These three groups of organic compounds are polycyclic aromatic compounds that can be generated during the combustion of organic material. Also, we have worked with brominated flame retardants that are used as flame retardants in different objects, as electronic devices, textiles, or plastics, and perfluorinated compounds that are used in coatings to produce water-resistant objects and also as part of firefighting flames. In the end, these compounds are released into the environment and can enter in the trophic chain, where they can be accumulated and finally can be a risk to consumers. For this reason, authorities have established control methods and legal or recommended concentration limits in fish and seafood products. From the three groups of contaminants studied in the project, only polycyclic aromatic hydrocarbons are regulated and maximum limit established in foods. However, new data are being reported about the occurrence and toxicity of the other groups, which will end with the establishment of new limits in the near future. So, our objective is to provide the food industry and also other type of end users with new tools to screen these compounds in a rapid and a cost-effective way. To accomplish this objective, we have developed an enzymatic biosensor. After the two previous presentations, you will probably be more familiar about what a biosensor is. In our case, instead of using an antibody as a bioreceptor, we have used enzymes that provided us with the required selectivity and sensitivity. As you will see, our methodology is able to detect several contaminants at the same time in a rapid way. In this slide, you can see the basis of our methodology. Target contaminants are able to inhibit the activity of a group of enzymes that have been selected for that purpose. In the absence of a contaminant or inhibitor, the enzyme catalyzes a reaction and transforms a substrate into a product that can be detected. When the contaminant is present, then the amount of product that is generated by the catalytic reaction of the enzyme is reduced. So, in the end, we can, by measuring the enzyme activity, we can detect the presence of a contaminant. Now, I would like to show you a video. So please, organizers, if you can launch the video, please. Okay, I will continue with the presentation. So the video have shown you the protocol for the analysis of polycyclic aromatic hydrocarbons, but the sampling protocol is pretty similar for the other group of compounds. Here you can see a brief summary. It consists of just wait the sample and make an extraction with an organic solvent, or adding several salts that they are called catchers, or in the case of perfluorinated compounds, we use a basic medium. Then centrifugate, take the supernatant and perform some cleaning steps. That here is the difference depending on the group of compounds, because they differ in their nature and chemical properties. Then, in the end, the extract is evaporated and reconstituted in the buffer solution. So this is the extraction protocol, and the assay is always the same. We should prepare a mixture of the enzyme. We add and incubate the enzyme with the extract for 10 minutes. And after that, we add the substrate to the mixture. We wait for 1 hour, and then we read the signal with a suitable reader. The last step of the methodology is the analysis and interpretation of the results. The signals recorded by the reader are proportional to the product concentration generated during the enzymatic reaction. As you can see in the figure, here the green signal is the buffer, the highest signal possible with this protocol. And here you can see the different signals obtained with the different samples that contains different concentrations of contaminants. By comparison, by comparing the signal from the sample with the blank sample, we can calculate a percentage of inhibition. During the validation of these assays, we have calculated a cutoff value for each contaminant that has been also correlated with a concentration. This value will allow the user of the kit to distinguish between a positive or a negative sample. In the case of a positive sample, when the contaminant is present above the concentration of interest, the assay will provide an inhibition higher than the cutoff. On the contrary, a negative sample, contaminants below the concentration of interest, will provide an inhibition lower than the cutoff. And these are a summary of the main results obtained with these assays for the three groups of contaminants. As you can see, in the case of polycyclic aromatic hydrocarbons, the limit of detections have been set at 2 ppbs and 10 ppbs, 2 ppbs of benzoapyrene and 10 ppbs for pH4, which is the sum of the 4 main hydrocarbons, and these are the limits established by the European Union regulation. We have estimated a cutoff value of 50%. Then, if the assay gives you values above 50% of inhibition, it means that polycyclic aromatic hydrocarbons are present in your sample above these concentrations. You can see there as well the numbers for the brominated flame retardants and for perfluorinated compounds. Just to conclude, the developed enzymatic bioassays allow the detection of hazardous contaminants present in the environment. The methods have been validated in salmon and sea bass, but they might be applied also to other seafood products or other presentations after checking the cutoff value. These fast screening methods can be used to reduce response time, reagents, and train labor during precise analysis. You will only, as you have seen in the video, you will only need a vortex centrifuge, an evaporator, and a reader, apart from common labware commonly used in laboratories. And as you have seen, it's quite simple. There are many steps, but they are mostly pipetting, diluting, mixing, and in the end, it can be done in a quite rapid way. It's important to say that when a sample is a positive result, subsequent analysis by chromatography or by a confirmatory method should be performed in order to identify the contaminant and the concentration, the exact concentration, identify the exact contaminant present in the sample. So just to conclude, I would like to thank Biorex also for their help and support to perform the video. And we have also, as Monica mentioned before, made a flyer that we can share with you as well. So thank you all for your attention. There you have my email if you want to contact me, or I'm now available to answer your questions.
Thank you for your presentation, Alex. But there is a question actually in the chat room. Which enzymes have you worked with? That's the question.
Enzymes from the family of cytochrome P4450. So we have, depending on the contaminant, we have select one or another. But the enzymes were from this family, were cytochromes.
Thank you for your answer. Any further question? Alex is online and ready to take any question you may have. I can't see any further question in the chat or any requests for the floor. So thank you very much to Alex, Monica and Katrina for their presentations on those fast screening methods. We will break shortly for 15 minutes and we will resume our work at 3:15. And we will be moving on to the the last two presentations in order to close off this first workshop. Thank you very much indeed. We meet again in 10 minutes.
Hello again. Let's now move on to our two final presentations on contaminants. I'll ask Amaya to talk about listeria. Amalia, please carry on and tell us about the activities you carried out on listeria. Where is your screen? Yes, seems to be working. Great, good. You can go ahead. Yes, yes. It's full screen.
Good afternoon to everybody. I'm going to talk about the bacteriophage biocontrol as an innovative solution to reduce Listeria monocytogenes from seafood products. Listeria monocytogenes is a well-known pathogen responsible for listeriosis, which is one of the most serious foodborne diseases worldwide. It has been reported an increasing trend in the number of listeriosis cases in the European Union during the last years, with more than 2,500 confirmed cases in 2018 and an associated deaths of almost 230, implying high mortality rates of more than the 15% in at-risk groups, such as elderly people, pregnant women, newborns, and immunocompromised patients. And seafood products have been associated with some listeriosis outbreaks over the time. In fact, the consumption of fish and fishery products, as well as crustaceans, shellfish, mollusks, and products thereof are the second and fourth most common causes of listeriosis in the list of strongly evidenced outbreaks that occurred in the European Union during the period 2010 and 2017. All this data underline the need to improve the control of Listeria monocytogenes in foods in general and in seafood in particular. Different studies have documented the presence of Listeria monocytogenes in fresh, minimally processed, and ready-to-eat seafood products from many countries during the last years. And the contamination of these products by Listeria monocytogenes typically occurs via contaminated raw materials or by cross-contamination during different steps of seafood processing. In addition, the ability of Listeria to forming biofilms on food contact surfaces, equipment, floors, and drains, together with its high tolerance to disinfectants, are determining factors in the great persistence of this bacteria even for years in food processing facilities. Furthermore, Listeria monocytogenes is able to survive and grow at a wide range of temperatures and pH values, high salt concentrations, and under osmotic pressures. So these unique abilities of Listeria monocytogenes make it very difficult to eliminate it from seafood processing equipments and environments, increasing thereby the risk of final seafood product contamination. Therefore, the control of Listeria monocytogenes occurrence in seafood products is an important challenge for the seafood industry and the public health sector. And this is particularly important for ready-to-eat seafood products, since they are consumed without further treatment or cooking. To prevent the occurrence of Listeria monocytogenes in food products, public health agencies and governments develop regulations of microbiological criteria for food products, including food safety criteria for Listeria monocytogenes. They also provide guidance to the food industry and support the research and development of innovative solutions with potential to be used as food safety control strategies. And one of these promising innovative solutions is the application of bacteriophages. But what are bacteriophages? For explaining a little bit what bacteriophages are and how do they work, we have prepared a short video. I will stop sharing my presentation in order to be able to launch the video.
Bacteriophages for seafood safety. The control of pathogenic bacteria causing foodborne diseases and the alarming emergence of antibiotic-resistant bacteria are important challenges for the food industry and the public health sector. In recent years, the so-called bacteriophage biocontrol is being increasingly accepted as an innovative, natural, and green food safety strategy to prevent the occurrence of non-desired bacteria in foods. But what are bacteriophages? Bacteriophages, or phages for short, are bacteria eaters, that is, viruses that naturally infect bacteria. Phages are the most abundant organisms on Earth. They are everywhere, even in the foods we eat. And why bacteriophages as food safety agents? Because phages exclusively infect and kill specific target bacteria, being innocuous to plants, animals, and humans. They are extremely specific. Each phage can typically infect one or few strains with a single species, and less frequently, few strains of different species within the same genus. So they do not affect other bacteria present in the food, the host, or the environment. Phages specifically recognize and attach to target bacteria. Then, they inject the genetic material and take control of the bacteria, which starts producing phage components. Rapidly, all new phages are assembled, and they produce an enzyme leading to bacteria lysis and the release of the new phages capable of repeating this lytic cycle again. As biocontrol agents, phages should infect as many target bacteria as possible. As they are very specific, the best strategy to achieve the broadest lytic spectrum is the formulation of phage cocktails with complementary phages, targeting diverse strains of different origins, antibiotic resistance, or even different species. Moreover, cocktails help to reduce the emergence of phage-resistant bacteria. Effective phage cocktails could be applied at different stages of the seafood chain to prevent the occurrence and growth of non-desired target bacteria. In aquatic environments, as an alternative to antibiotics to control infections during seafood production. and in food products and food processing environments during seafood processing. Bacteriophage biocontrol could contribute as natural and green strategy to ensure the safety of seafood products.
Okay, I will share my presentation again. So in this context, one objective of the Seafood Tomorrow project, as has been commented before by Antonio, was to implement innovative strategies to improve the safety of seafood products. And among them, ASTI, in collaboration with the industrial partner Phage Technology Center, have worked together to develop a bacteriophage-based product as an efficient strategy to control Listeria monocytogenes in seafood. For this purpose, the first step is usually the isolation of different, in this case, Listeria-specific bacteriophages, also called Listeriaphages, from food and environmental sources, and by using different Listeria strains as target host. In our case, as the starting point within the Seafood Tomorrow project, we selected 16 Listeriaphages from a large, previously isolated collection of more than 75 phages. And then we had to characterize them. Why? Because not all phages are suitable for food applications. Listeria-specific bacteriophages must fulfill several requirements to be used as biocontrol agents for food safety applications. The most important criteria are based on their safety, specificity, efficacy, and stability. So the 16 selected Listeriaphages were characterized firstly morphologically, as you can see in these two photographs. All phages showed a similar structure with an icosahedral head containing DNA and a long tail with tail fibers at the end. And considering their morphological characteristics as well as their genome size, 10 phages were classified within the family Siphoviridae, whereas the other 6 phages were classified within the Myoviridae subfamily with larger genome size but shorter tails. Regarding their safety, the de novo next generation sequencing of the whole genomes of selected bacteriophages was done. The bioinformatic analysis of the raw genome sequences showed that all these 16 phages were genetically different among them. However, 3 of them showed lysogenic conversion, so they were discarded for food applications, while the other 13 phages were confirmed as lytic phages able to display the lytic cycle explained before in the video that causes the lysis and death of the target Listeria cells and the release of the new phages able to infect other Listeria and repeat this lytic cycle again. Therefore, these 13 lytic phages could be considered safe for their use as biocontrol tools in food applications and were further characterized regarding their specificity and efficacy. Phages were characterized by determining the lytic activity of each individual Listeria phages against a diverse panel of near 50 different Listeria monocytogenes strains, including both collection and wild-type strains from different serotypes and origins, including seafood isolates, and not only Listeria monocytogenes strains, but also other non-monocytogenes Listeria strains reflecting the environment under study. As explained in the video, achieving a broad athletic spectrum with a single phage is very difficult, since bacteriophages are highly specific against their target strains. However, this results on the specificity of individual Listeria phages allowed us to design different phage cocktails, different mixtures of phages combining from 3 to 7 complementary Listeria phages. And we also analyzed the lytic activity of these cocktails, showing that they were effective against a broader range of target Listeria strains than the application of individual phages. At this point, we selected the most 2 effective cocktails, showing the broadest lytic spectrum and being able to lyse more than the 90% of tested Listeria strains. Design phage cocktails should also remain stable and effective under different storage and application conditions until they reach and infect their target Listeria monocytogenes cells within the seafood products. Therefore, in this point, we also characterize their stability and effectiveness in a wide range of pH values and temperature. Results showed that phages induced that phages included in both cocktails were stable and effective for at least 4 months under storage and application conditions of temperatures from 4 to 25 degrees and pH values ranging from 2 to 9.5. And finally, once characterize the most relevant technological properties for food safety applications of the defined Listeria phage cocktails, in order to be able to define the final product, we worked on the assessment and optimization of the effectiveness of these cocktails for the reduction of Listeria monocytogenes on the surface of contaminated seafood products. The effect of, we studied the effect of several variables, such as the type of fish product or the product area, the load of Listeria monocytogenes, the amount of phages used for treatment, or the mode and time point of application. And in order to select the most efficient cocktail formulation and also the most efficient treatment conditions. And these most effective cocktail and treatment conditions were finally validated in raw and ready-to-eat seafood products in order to allow definitive conclusions on the efficacy in reducing, in this case, naturally occurring low Listeria monocytogenes counts all over the storage period of the fish products. And we obtained very good results, showing that Listeria phage cocktail was able to reduce the initial load of Listeria monocytogenes while maintaining it below the legal limit of 100 colony forming units per gram at the end of the shelf life of both raw and cold smoked salmon. So, and as a final step to put on the market a new phage-based product for food applications, Listeria phages should be able to be produced at large commercial industrial scale. And to address this point, the industrial partner PTC optimized many critical parameters of the production and purification process in order to allow the scale up of the production of stable phages at optimal yields from small volumes at lab scale to medium volumes at pilot scale, and finally to industrial commercial scale volumes of up to 100 liters. So, in summary, we can conclude that biological properties of lytic bacteriophages, as well as the new data obtained within the Seafood Tomorrow project on the effectiveness of Listeria-specific bacteriophages on seafood products, make bacteriophage biocontrol a promising strategy for seafood safety applications. In fact, the use of Listeria bacteriophages could contribute as an additional innovative solution to control the prevalence of Listeria monocytogenes in seafood. That's all. Thank you very much for your attention.
My question, it's maybe a naive question, but I would like to know if I would like to know if do you think that your work in the project could be influence the regulation about the use of phages? Because I think that we cannot use for the moment phages pour lutter contre les Listeria.
To fight it.
Okay. Yes, you are true. There is no specific regulation. There is no clear path regarding how to register a bacteriophage product in the European Union. The EFSA panel, the EFSA has published different scientific opinions related to the use of bacteriophages, but the European Commission's in 2018, if I don't remember wrong, did not agree on a regulatory path for bacteriophages in food production and relegated the responsibility back to the individual member states. So it depends on the country on how to manage bacteriophage application as in food or for food applications. For example, there are some countries such as Switzerland or non-European countries, but such Switzerland or Israel, Canada, Australia, New Zealand, or Brazil that have already approved the application bacteriophage applications on foods, and of course in the in the USA there are different bacteriophage-based products for food applications with the GRAS generally recognized as safe status. But let's see. I hope that that this data obtained with the Seafood Tomorrow can help to accelerate this regulatory status or regulatory approval of bacteriophages for food safety applications.
Thank you very much indeed, Maya, for your answer and also for the very clear presentation. Thank you very much indeed. And I was looking at the chat again to see whether we had another question, but it's not the case. So thank you very much indeed, Maya. And may I ask, Andy, are you on the call? If that's the case, can you click on the request to speak button to address the meeting on this matter of the detection of norovirus?
Afternoon, everybody. My name's Andy Younger. As you've just heard, I'm going to talk about norovirus depuration from shellfish. Share the right screen. Can you see that okay? Can everybody see that?
Yes, no problem at all. We can see your presentation. Maybe you can put it on presentation mode and so that we get the full screen if you manage to do so.
Yes, afternoon everyone. I'm Andy Younger from CFAS in Weymouth, which is in the UK. No longer part of Europe. Apologies for that. So the introduction really is, as I say, depuration, which is effectively the treatment process for Class B shellfish under European regulations. So what we're talking about is or tanks of seawater. It's designed to remove microbiological contamination from and it's very good at removing bacteria. So E. coli is very quickly removed, but it's not so good at removing viruses. And in particular, norovirus, which causes most illness really associated with shellfish consumption across Europe and across the world, in fact. So what we do know is depurated shellfish can be free from E. coli, but can still be associated with norovirus outbreaks. We did a study back in 2013 looking at norovirus depuration from oysters. This was only using PCR, norovirus by PCR, bacteriophage by viability. So if you look at the graph here, you'll see the green lines are norovirus depuration. The axis, the y-axis is percentage remaining. The x-axis is days of depuration. So after 2 days, for example, if you look at the red lines, that is E. coli removal, and you'll see after 2 days, it's all gone at both temperatures 8 and 16 degrees in this case. If you look at the yellow lines, that is FRNA bacteriophage. You've heard about bacteriophages just from Maya. This particular one is always present in sewage. It's a virus very similar to norovirus, but the advantage of this particular one is we can grow it so we know when we've got viable infectious virus. The problem with norovirus is we can't do that at the moment in a routine testing way, so we have to rely on PCR testing. So if you look at the bacteriophage, the yellow line, you'll see that it takes a lot longer to remove than E. coli, but after 14 days, certainly at 16 degrees, it has finally gone. But in terms of norovirus, there's not a lot of removal at all, perhaps a little bit more at 16. What we don't know, or what we didn't know, is how much of that virus is viable. So part of this study was to use bacteriophages to try and make some assessment. So as I say, the challenge was the current routine detection methods for norovirus, not virus or virus infectivity. That, of course, is what represents the risk to the consumer. So the use of surrogate viruses, such as RNA bacteriophage, or FRNA, as I will refer to it because that's less of a mouthful, that can be used to indicate risk in some situations. Now, the advantage of using FRNA phage is, as I've said, it's easy to grow. So we can actually quantify the viability and the infectivity of the virus. And obviously, enhanced depuration procedures based on norovirus removal and infectivity are used for the shellfish industry. So that was the whole idea of this project, particularly during the winter months when we see most norovirus presence in the community, certainly in temperate climates in northern Europe. So FRNA phage, as I say, we measured that. We test for it with a standard plate viability assay. And that was looking specifically at GA, which is one of the genotypes or strains that we find in sewage. There are 4 different genotypes or groups of FRNA phage, and only 2 of those generally found in human waste, sewage, and GA is one of those. So that's the one we looked at, both in the viability test and with using the PCR method. So we could directly compare how much genetic material we got there, how much phage generally was there, and how much of that was actually viable. So the methods, as I say, we looked at Pacific oysters, Crassostia gigas. That was environmentally contaminated, naturally contaminated shellfish. We looked at various factors. Water temperature was the main one, 18 degrees versus 8 degrees. We looked at feeding, microalgae that we grew in the lab versus diet. We looked at light and dark regimes, disturbance versus non-disturbance, and by that I mean the vibration caused by the pumps which are attached to the side of the tanks, generally speaking, versus tanks that didn't have any vibration at all. And we looked at continuous flow through of water versus recirculated, unfiltered seawater. And we tested the shellfish for E. coli, norovirus by PCR, as I said, looking for genogroups 1 and 2, which are those associated with human illness. And we also looked at FRNA phage by culture, as I say, and FRNA phage by PCR. And that was looking for the GA. Shellfish we placed with the help of a local operator near a sewage discharge. Sewer overflow capacity as well. So we were expecting that to provide quite. We used commercially available tanks, relatively small compared to what are used in Europe, I know, but these are quite common in the UK, these particular tanks. Litres, made by Marine Centre, a company in the UK. We looked at, so there's one particular trial we had to use where we needed seawater supply, so we had to use and all water was recirculated via 2 25-watt UV units, and the oysters were held in standard Alibert trays. This is a side profile of the tanks. So basically, the water recirculated, entered the tank via a spray bar, which causes aeration of water for oxygen. The shellfish in the trays, water taken out by the suction bar, and then recirculated via the UV unit. Now, because we were using naturally contaminated shellfish, we had to rely on a relatively small window, the winter window for norovirus being high in the shellfish. So that provided a bit of a limited opportunity for us to carry out the trials. We had to get on with it, basically. Now, as luck or otherwise would have it, unfortunately, the year we tried these trials in 2018-19, the incidence of community, this is the black line, the lower incidence is much lower than we normally expect. So that did make the trials much more difficult to get useful results from. to the point where when we came to the final validation trial, the results.
So what I was saying was effectively that unfortunately, because the levels pre-deprivation getting towards the end of the norovirus season were quite low, the results in the last trial, which was the big validation trial, weren't really of any use. The results were too low. So, which was a shame because it was an expensive trial to undertake. But there we are. That's the way science works sometimes. But the results from the temperature trials were the most promising. This graph here shows percent organisms remaining on the left-hand side, the y-axis, and along the bottom we have the time of deprivation from 0 to 5 days. So 3 data points: 0 days, 2.5 days, and 5 days through deprivation. And what we can see is the top blue lines, dotted line is 18 degrees. The continuous line is 8 degrees. So what it shows is we had minimal removal of norovirus genogroup 1. But if you look at the grey lines, we had much better removal, and that was norovirus genogroup 2. And particularly at 18 degrees, the dotted line, we saw quite significant removal. In fact, 60% removed after 5 days. Now the RNA phage results were a little bit confusing. We can see the red line here changing between the 18 and 8 degrees. So, but essentially, phage, both by viability test and by PCR, seemed to be removed more readily, more quickly than both genotypes for norovirus. So that was a bit unfortunate, and we were hoping that FRNA phage would be a good indicator of virus contamination, norovirus contamination in deprivation. But the fact it was removed more quickly would suggest that maybe in this instance, at least, it wasn't. But we will be doing some further work on that, which I'll mention a bit later. And as expected, E. coli removal was very good. And that's the black lines, the black solid line and the dotted black line. So these are the results plotted using regression lines, taking account of the variability. And again, it just reinforces the conclusions that norovirus genogroup 1 was barely removed. I think about 16% removal after 5 days, whereas we had much better removal. The dashed lines here, the blue and red dashed lines for 8 and 18 degrees. So the dotted lines, blue and red at 8 and 18 degrees for norovirus genogroup 1. But again, showing the dashed lines here, the blue and red lines, showing that FRNA phage was more readily removed than the norovirus. So it's good in terms of norovirus removal, particularly for genogroup 2, but the FRNA phage as an indicator would appear to be a bit disappointing. So the conclusions from the study, we found that deprivation was twice as effective at 18 degrees than compared with 8 degrees after 5 days. And norovirus genogroup 2 was consistently better removed than norovirus genogroup 1. That may be down to binding of genogroup 1 to the oyster gut, which has been found by some workers at Ifremer in France. Norovirus genogroup 1 removals were negligible. As I say, I think the best we had was 16% removal after 5 days at 18 degrees. Genogroup 2 levels were reduced up to 60%, indicating that commercial purification can be useful for some strains of the norovirus. And as I say, you do tend to see quite a lot of norovirus genogroup 2 strains in community outbreaks in the UK and I believe across Europe. On the salinity trials, we did see that an effect which suggested that you do need to match the salinity as closely as possible to the environment to which the shellfish have become acclimatized. So plus or minus 20% of the salinity in their production areas will give you the best results. And the other factors we trialed, which included feeding, didn't have a significant improved effect at all on norovirus removal, which came as a surprise, really. We did think that feeding would help, but it didn't show any. In fact, it almost seemed to be the other way around, that lack of feeding seemed to increase norovirus removal, if anything. And because FRNA phage was better removed in our study than the norovirus genotypes, that would suggest that it's not a good indicator of norovirus for deprivation. The whole idea of an indicator is that it's not removed as quickly so that you're having a safety margin there. But we will be looking at this in more detail. So summary outputs, we've tested a method, elevated temperature in particular, that lowers norovirus levels from contaminated Class B shellfish, oysters. I should say there might be a difference between different species, so these results only apply to oysters. And this is obviously from areas experiencing a temperate climate, similar to the UK. So France, northern France in particular. And our method may reduce health risks by improving seafood safety, and seafood processors and producers can use this method for the benefit of consumers worldwide. We've delivered a protocol for the industry that's available on Zenodo. We've also published a paper in the Food and Chemical Toxicology journal. Again, that is available free of charge, free access on Zenodo. And I mentioned ongoing work. We've been invited to take part or to assist with the Oxivir 2 project. That was invited. We received an invitation from the National Shellfish Farming Committee in France, looking at the use of bacteriophage for investigating norovirus infectivity. So, as I say, our results were a bit inconclusive, and we would hope to provide the benefit of our experience in fine-tuning this particular project. That's something that's going to be EMF funded, and hopefully start imminently. So that's all from me. Apologies for all the technical difficulties. I hope you got something from that. If there's any questions, then please let me know. Thank you.
Okay, thank you, Andy. I don't know if you see, but you have a question on the chat. Would you like to see on it, or maybe I can read it? But
oh yeah, is this instead of doing field trials and waiting for norovirus levels to reach desired levels, is it dangerous to just artificially infect bivalves in closed environment and to observe norovirus removal post depuration? Yeah, no, we have tried this with mixed results previously. The results I showed you from 2013, they were done using artificial contamination. What we did find was that the results levels seemed to go up even after we'd finished dosing the shellfish, and so it did make interpretation of results during the depuration more difficult, really. It's also getting hold of norovirus material that can be quite difficult. We can obviously use sewage. That's one thing we've tried with mixed success. We have in the past used clinical samples of feces to dose shellfish, but that can be quite difficult to get hold of. So yes, you can, but artificially infecting is more difficult, and arguably it's not necessarily representative of the real world situation, which is why we went for natural dosing. But it was just unfortunate in our particular case that the results, the levels in the environment that particular year weren't very high. I think whichever way you go, there are going to be difficulties, and it's just a question of trying to do the best you can within the the limitations of the methodology that you choose. But it's a good question. Yeah.
No further question. Thank you very much for your presentation. And you were the last presenter for this workshop, and I would like to ask our director, the Aquima director, to say a few words in order to close the workshop. And of course, we'll go back to Antonio for the final word. Good afternoon.
It's been a long day. So 35 partners, 19 European countries, 7.5 million euros, 3.5 years. This was a large project. I'd like to take this opportunity to thank all the project partners and all the speakers today, and the audience, those who asked questions, and the coordinator will have the final word. So tomorrow's seafood will always be a topical issue. You will always have to think about tomorrow, and this issue requires mobilizing several fields which are related to one another. Meeting customers' expectations also means meeting the customer expectations for pleasurable products. You shouldn't neglect that aspect, but we should also meet the consumers concerned to consume safe products, healthy products, whilst reconciling this with sustainable development requirements. It also means that you have to talk to companies, medium-sized companies, but also micro companies in the seafood processing sector. They have to modernize their processes to meet all these objectives. All these topics are topics which have kept us busy at Aquamer for several years. The relevance of this consortium around this project was to create communities around issues and themes. And when it's necessary, we will be able to start new projects, because this Seafood Tomorrow project opens up new perspectives with new topics and new projects. So we will be approaching you again because we monitor all upcoming European projects. And since we know each other, we've been working together, we'll approach you for new projects. I won't take any more of your time, and I give the final word to the project coordinator. Thank you. Mr. Missoni. Antonio, are you here for the closing words of this webinar? Yes, here I am. Here I am.
Again, well, I would like to say some words. First of all, to the persons, and particularly to the companies that have been involved in this workshop. I hope that the session that you have heard today have opened new doors in terms, and it will be very helpful also for the French food, seafood production and processing sectors. There was a lot of a diversity of tools, not all were covered. I'm sure that the doors are opened for the exploitation of these tools, that you have seen that they were all validated. And please ensure with the presenters that are certainly the best players to disclose the tools. Please make sure that you utilize these new solutions for the production and processing of seafood in the future. Please liaise with them. As Julie has mentioned, well, the presentations will be made available, as well as the recorded workshop, which is important for you to. There was a lot of information, so that you can come back and trace the relevant information for you. I would like to congratulate all partners. You have seen that the presentations were excellent. They were clear, constructive, and they were objective. First of all, we have to bear in mind that for the last year, all these partners, they were like all of us, they were able to work in particularly difficult conditions with confinements, etc. And they were able to successfully accomplish the objectives of the project. And this is really nice to see that almost at the end of this project, we have almost 100% of the objectives accomplished. This is excellent. I would like to say also a special thank to Aquamer for the organization, particularly. It was very nice. Well, there was a lot of challenges, not only for you, but also for the speakers in terms of the new technologies that are involved with these online workshops. But I think there was an excellent organization and a clear and a good tool to be used in the future for sure. And I would like at last to invite you all to see the final results being disclosed in the project website. And please contact all the partners that you consider that the solutions are relevant for your companies. Thank you very much, Julie and Aquimer, for this excellent organization, and to the speakers.
Thank you, Antonio. Thank you to all the participants, and hopefully see you very soon. Bye-bye.